In 2004, the word designer baby became a headline and a common word in science fiction media. The term is defined as a human embryo that has been genetically modified. More specifically, I will be discussing germline modification, which is a form of genetic engineering that changes reproductive cells. This type of engineering is inheritable, meaning the modified genes will be carried to the following generations. Most of the science behind designer babies has to do with CRISPR, which was explained in the previous presentation. There are tools and techniques that are specific to the testing and research of designer babies. Most germline modification techniques are based on the concept of transferring DNA molecules known as transgenes to female-derived cells, especially a zygote. When the transgenes are introduced into the nuclear environment of the zygote, it usually integrates into the cell's endogenous DNA, therefore adding new genetic sequences to the host genome. The transgene sequence for the most part is stable and becomes a permanent feature of the modified host genome. Germline editing has been around for years, mostly used in animals. Therefore, there are many forms of germline editing. The Archives of Medical Research website lists these forms. The most used and established method for delivering transgenes is pronuclear microinjection. Typically, with PMI under a microscope, a fine glass needle is used to insert a nanoliter of DNA into the one zygote. PMI can be used to deliver large transgenes such as human genes. This method has been used to transfer entire artificial chromosomes. Here is a demonstration by Sigma Aldrich that is conducted using mice embryos. The holding pipette and injection needle are on opposite sides of the embryo. The holding pipette ensures that the embryo is stationary while the injection needle is inserted. The tools to perform PMI have roughly stayed the same for about 50 years and look something like this. This was not a very effective technique, as cells would die 40% of the time due to the injection needle causing swelling or puncturing the cell. Technology around germline modification is rapidly growing. We are constantly making new steps in the direction of designer babies. In 2014, BYU created a new technology called a nano-injector device.
We've developed a new technology which we call nano-injection that allows us to move DNA into cells. The current problem with microinjection is that the microinjection needle, which is the hollow needle to inject the DNA, it is large and it can damage the cell as it actually goes in. So you see a lot of loss of cell life. Because of the engineering, we now have that capability to make really, really small needles. In fact, they're so small, we actually call them lances. So the lance is different than the microinjection needle because the lance is solid. So what's really cool about the technology is that DNA is a negatively charged molecule. And so we could put a positive charge on the lance and DNA will actually collect on the outside of it. And then we could put that lance into a cell and reverse the charge and it drops DNA off there. This has huge advantages. The MEMS system is small enough that we can actually make the lance 10 times smaller than a microinjection needle. And the great advantage of having something smaller is that it will have much less damage as you actually insert it into the egg. It's amazing that we can make something so small. The nanoinjection lance is about 100th the size of a human hair. Here you can see how tiny the chip is when you put a pen next to it. And that chip is the one with about 12 to 20 different nanoinjectors on it. There are several applications that will help this to transform the industry. It does give us a new way to actually manipulate DNA to move it to places that we want it. We also can look at genetic disorders, so genes that are defective and how to actually correct those. And this is all because of this new technology.